We welcome our listeners today to the session on co- and post-translation modifications and inhibitors. We start with the first half of the session, that is co- and post-translation modifications. Translation is the process that takes the information passed from DNA as messenger RNA, abbreviated as mRNA, and turns it into the sequence of amino acids of the proteins. The mRNA possesses 64 types of triplet codons. The process of translation occurs in the following steps. Step 1. Activation of amino acids. Activation of amino acids is brought about by the binding of an amino acid with adenosine triphosphate, abbreviated as ATP, in the presence of activating enzymes known as amino acyl ribonucleic acid synthetases. This reaction results in the formation of amino acyl adenosine monophosphate, abbreviated as AMP, enzyme complex as shown here on the screen. The amino acyl RNA synthetases are amino acid specific, that is, for different amino acids, different enzymes are required. These enzymes can recognize the respective transfer RNAs, abbreviated as tRNAs also. Step 2. Transfer of amino acids to tRNA. The complex, that is amino acyl AMP enzyme complex, formed in the preceding step reacts with a particular tRNA and transfers the amino acid to the tRNA to form amino acyl tRNA complex as displayed here on the screen. The AMP and enzyme are liberated. The tRNAs are also amino acid specific. It means at least 20 types of tRNAs are required. It is now well known that one type of amino acid can be carried by more than one type of tRNA, but one type of tRNA cannot carry more than one type of amino acid. Step 3. Initiation of polypeptide synthesis. For the initiation of polypeptide synthesis, certain initiation factors, abbreviated as IF, are required. In prokaryotes, these factors are of three types: IF1, IF2 and IF3. They are loosely associated with the 30S subunits of ribosomes. IF3 is required for the binding of 30S subunit of ribosomes to the mRNA starting site. It also functions as a ribosome dissociation factor. IF2 binds the initiator F-met tRNA and carries it to the ribosome together with guanosine triphosphate, abbreviated as GTP, in response to the first AUG codon. IF1 participates in the interaction between IF2 and the initiator tRNA. During protein synthesis, once the large ribosomal subunit binds to the small subunit, the initiation factors are released and can be reutilized. Initiation process in prokaryotes takes place as follows. In Escherichia coli, abbreviated as E. coli, there are two tRNAs for methionine. They are tRNA M-met, that is non-formilatable, and tRNA F-met, that is formilatable. The later one deposits methionine at first place, whereas the former deposits methionine at the intercalary positions only. Methionine binds with tRNA F-met to form met tRNA F-met complex. Met tRNA F-met complex undergoes formylation in the presence of transformylase enzyme and a format source to yield F-met tRNA F-met complex. IF3 factor binds to 30S subunit and produces IF3 30S complex. IF3 30S complex then binds to mRNA to form IF3 30S mRNA complex. The 30S subunit binds with the first AUG codon, that is initiation codon of the message on mRNA. The recognition of this particular AUG message results from the interaction between complementary nucleotides of the 3' end of the 16S ribosomal RNA, abbreviated as rRNA, of the 30S subunit with a region of the message just before the initiation codon. The mRNA is read in a 5' to 3' direction. The binding of the 30S subunit to the mRNA requires the factor IF3 and the interaction seems to be stabilized by IF1. The ribosomes have two sites for association with tRNA. They are amino acyl, that is A site, and peptidile, that is P site. The A site is generally the one in which amino acyl tRNAs enter the ribosome mRNA complex, while P site is the one from which the tRNA leaves after having donated its amino acid. In the case of first amino acyl tRNA, that of F-met tRNA F-met, the entry appears to be directly into the P site of the 30S subunit. GTP is also required for the successful initiation. GTP binds to the F-met tRNA F-met before it attaches to the mRNA 30S complex. Once the initiator tRNA becomes attached, IF3 is released and the 50S ribosomal subunit attaches to the 30S subunit to form the complete initiation complex, that is 70S mRNA F-met tRNA F-met. At this point, GTP is hydrolyzed and the GDP, IF1 and IF2 are released, and the stage is now set for the elongation step in prokaryotes. Initiation in eukaryotes is of two types, that is cap dependent and cap independent initiation. In eukaryotes, at least 10 soluble protein factors have been identified. We will now learn cap dependent initiation in eukaryotes. The eukaryotic initiation factor 3, that is EIF3, is associated with the small ribosomal subunit and it plays a role in keeping the large ribosomal subunit away from premature binding. EIF3 also interacts with the EIF4F complex, which consists of three other initiation factors, that are EIF4A, EIF4E and EIF4G. EIF4G is a scaffolding protein that directly associates with both EIF3 and the other two components. EIF4E is the cap binding protein. It is the rate limiting step of cap dependent initiation and is often cleaved from the complex by some viral proteases to limit the cell's ability to translate its own transcripts. This is a method of hijacking the host machinery in favor of the viral, that is cap independent messages. EIF4A is an ATP dependent RNA helicase which aids the ribosome in resolving certain secondary structures formed by the mRNA transcript. There is another protein associated with the EIF4F complex called the poly A binding protein, abbreviated as PABP, which binds the poly A tail of most eukaryotic mRNA molecules. This protein has been implicated in playing a role in circularization of the mRNA during translation. This pre-initiation complex, that is 43S subunit or the 40S and tRNA, accompanied by the protein factors, move along the mRNA chain towards its 3' end, scanning for the start codon, typically AUG, on the mRNA, which indicates where the mRNA begins coding for the protein. In eukaryotes and archaea, the amino acid encoded by the start codon is methionine. The initiator tRNA charged with met forms part of the ribosomal complex, and thus all proteins start with this amino acid. The met charged initiator tRNA is brought to the P site of the small ribosomal subunit by EIF2. It hydrolyzes GTP and signals for the dissociation of several factors from the small ribosomal subunit, which results in the association of the large subunit or the 60S subunit. The complete ribosome, that is ATS, then commences translation elongation, during which the sequence between the start and stop codons is translated from mRNA into an amino acid sequence. Thus, a protein is synthesized. Cap independent initiation. The best studied example of the cap independent mode of translation initiation in eukaryotes is the internal ribosome entry site, abbreviated as IRES approach. What differentiates cap independent translation from cap dependent translation is that cap independent translation does not require the ribosome to start scanning from the 5' end of the mRNA cap until the start codon. The ribosome can be trafficked to the start site by IRES transacting factors, abbreviated as ITAFs, bypassing the need to scan from the 5' untranslated region, abbreviated as UTR. This method of translation has been recently discovered and has found importance in conditions that require the translation of specific mRNAs despite cellular stress or the inability to translate most mRNAs. Moving ahead, we now discuss step 4, elongation of polypeptide. After the formation of 70S mRNA Fmet tRNA Fmet complex in prokaryotes, elongation of polypeptide chain is brought about by the regular addition of amino acids. The elongation process requires the presence of factors Tu, Ts and G. The first step in elongation involves the entry of the second amino acyl tRNA to the A site of the ribosome. This and the subsequent amino acyl tRNA combine with Tu factor and GTP before binding to the A site to form Tu GTP AA tRNA complex. Tu factor probably helps in codon recognition and for orientation of the incoming amino acyl tRNA. After the attachment of Tu GTP AA tRNA complex to the ribosome, GTP is hydrolyzed in the presence of GTPase and a Tu GDP complex is released. Ts factor rejuvenates the Tu GTP. A peptide bond is formed between first and the second amino acids. This bond is formed between the free carboxyl group of the peptidyl tRNA at the P site and the free amino group present with the amino acyl tRNA at the A site. The enzyme involved in this reaction is peptidyl transferase, and it is an integral part of 50S ribosomal unit. After the formation of peptide bond, the tRNA at P site is deacylated and the tRNA at A site now carries the dipeptide or the polypeptide. The tRNA from P site is dislodged. The ribosome moves one codon along the mRNA in the 3' direction. It is termed as translocation and it is accompanied by the movement of the tRNA dipeptide to the P site of the ribosome, still hydrogen bonded to the second codon of the mRNA. The translocation requires G factor, also termed as translocase, and hydrolysis of GTP. When the tRNA with its attached dipeptide moves to the P site, the A site is open to the entry of another amino acyl tRNA, in this case one whose anticodon is complementary to the third codon. A peptide bond is formed between second and third amino acid and the cycle is repeated as discussed. As the ribosome continues to move along the mRNA, the growing polypeptide chain termed the nascent polypeptide increases in length. The direction of elongation is from amino to carboxylic group. In eukaryotes, elongation depends on eukaryotic elongation factors. At the end of the initiation step, the mRNA is positioned so that the next codon can be translated during the elongation stage of protein synthesis. The initiator tRNA occupies the P site in the ribosome and the A site is ready to receive an amino acyl tRNA. During chain elongation, each additional amino acid is added to the nascent polypeptide chain in a 3-step microcycle. The steps in this microcycle are positioning the correct amino acyl tRNA in the A site of the ribosome, forming the peptide bond and shifting the mRNA by one codon relative to the ribosome. Translation can also be affected by ribosomal pausing, which can trigger endonucleolytic attack of the mRNA, a process termed mRNA no-go decay. Ribosomal pausing also aids co-translational folding of the nascent polypeptide on the ribosome and delays protein translation while it is encoding mRNA. This can trigger ribosomal frame shifting. Step 5. Termination of polypeptide. The termination of polypeptide chain in both prokaryotes and eukaryotes is brought about by the presence of any of the 3 termination codons, namely UAA, UAG and UGA. There are no tRNAs with anticodons complementary to these triplets. When the ribosome reaches one of these termination codons, the signals are received to stop further elongation and terminate the polypeptide. The termination involves release factors that are RF1, RF2 and RF3 and GTP. The RF1 and RF2 help in the recognition of the termination codons, whereas RF3 or S is involved in the release of the completed polypeptide from its attachment at its C-terminal end to the last tRNA that had entered. After this process, the ribosome separates from mRNA and dissociates once again into its subunits in the presence of IF3. it has been revealed that more than one ribosome is involved at one time in the translation of a single message. During translation, a number of ribosomes attach along the length of mRNA. This complex of ribosomes and mRNA is termed as polyribosome or polysome. Each one of the ribosomes initially attaches to the mRNA at the initiation codon and then moves from that point toward the 3' end. As soon as a recently attached ribosome moves a sufficient distance along mRNA, the next ribosome becomes associated with the mRNA and begins to translate the message simultaneously with its translation by previously attached ribosomes. The simultaneous translation of the same mRNA by several ribosomes greatly increases the potential for protein synthesis. Step 6. Modification of released polypeptide. The released polypeptide is deformylated in the presence of an enzyme deformylase, which removes the formyl group of first amino acid methionine. Due to action of some exopeptidases, amino acids are removed from either the N-terminal end or the C-terminal end or both. The polypeptide chains, singly or in association with other chains, become folded and form tertiary structures. Further, we talk about the post-translation modifications. The newly synthesized protein is modified to become functionally active. The various post-translation modifications are: number 1, trimming. It removes excess amino acids. Number 2, covalent alterations. Covalent alterations further includes phosphorylation that may activate or inactivate the protein. Glycosylation, it targets a protein to become a part of the plasma membrane or lysosomes or be secreted out of the cell. Hydroxylation, as seen in collagen, is required for acquiring the three-dimensional structure and for impairing strength. Gamma carboxylation and isoprenylation. Number 3, protein degradation. Defective or destined proteins for turnover are marked for destruction by attachment of ubiquitin protein. Proteins marked in this way are degraded by a cellular component known as proteasome. Now, we will discuss the second half of the topic, that is inhibitors. A protein synthesis inhibitor is a substance that stops or slows the growth or proliferation of cells by disrupting the processes that lead directly to the generation of new proteins. Protein synthesis inhibitors are an extremely diverse and ubiquitous class of biochemical molecules. Their function ranges from bacterial translation inhibition to blocking synthesis of specific amino acid residues necessary for the production of a protein. In general, antibiotics are biochemically or fungally produced substances that inhibit the growth of other organisms. Most antibiotics, like many pharmaceuticals, block translation in protein synthesis. These substances are effective because they take advantage of the tremendous complexity involved in the synthesis of proteins. The following are some of the synthesis inhibitors and the stages which they target. At early stages, rifamycin, rifampin inhibits prokaryotic DNA transcription into mRNA by inhibiting DNA dependent RNA polymerase by binding to its beta subunit. Linezolid acts at the initiation stage by preventing the formation of the initiation complex. Tetracyclines and tigecycline block the A site on the ribosome by preventing the binding of amino acyl tRNAs. Aminoglycosides interfere with the proofreading process, causing increased rate of error in synthesis with premature termination. Chloramphenicol blocks the peptidyl transfer step of elongation on the 50S ribosomal subunit in both bacteria and mitochondria. Macrolides bind to the 50S ribosomal subunits and inhibit peptidyl transfer. Quinopristine, delphopristine act synergistically to inhibit peptidyl transfer. Quinopristine binds to a nearby site on the 50S ribosomal subunit and prevents elongation of the polypeptide and enhances the release of incomplete chains. Fusidic acid prevents the turnover of elongation factor G, that is EFG, from the ribosome. Puromycin has a structure similar to that of the tyrosinyl amino acyl tRNA. Thus, it binds to the ribosomal A site and participates in peptide bond formation, producing peptidyl puromycin. However, it does not engage in translocation and quickly dissociates from the ribosome, causing premature termination of polypeptide synthesis. Streptogramins cause premature release of the peptide chain. Retapamulin and mupirocin also are protein synthesis inhibitors of unspecified mechanism. Chloramphenicol, erythromycin, clindamycin, linezolid, telithromycin, streptogramins and retapamulin are the antibiotics that act at the binding site of the 50S ribosomal subunit and blocks peptidyl transferase activity. Penicillin inhibits transpeptidation enzymes involved in the cross-linking of polysaccharide chains of the bacterial cell wall peptidoglycan. Cycloheximide inhibits translocation of peptidyl tRNA. Becitran inhibits cell wall synthesis by interfering with the action of the lipid carrier that transports wall precursors across plasma membrane. Thiostrepton inhibits ribosome dependent EFTU and EFG GTPase. Sulfonamides inhibit folic acid synthesis by competition with p-aminobenzoic acid. Streptomycin causes codon misreading, insertion of improper amino acid and prevents formation of initiation complex. It is important to note that protein synthesis in eukaryotes and prokaryotes proceeds through the same steps of initiation, elongation and termination. These general similarities in mechanism allow certain antibiotics to act on both systems effectively. Summary: translation is the synthesis of a polypeptide using the information encoded in an mRNA molecule. The process involves mRNA, tRNA and ribosomes. tRNA has a unique structure that exposes an anticodon which binds to codons in an mRNA and an opposite end that binds to a specific amino acid. Binding of an amino acid to a tRNA is carried out by an enzyme called amino acyl tRNA synthetase. Translation consists of three basic steps that are initiation, elongation and termination. Initiation involves the formation of the ribosome or mRNA or initiator tRNA complex. Elongation is the actual synthesis of the polypeptide chain by formation of peptide bonds between amino acids. Termination dissociates the translation complex and releases the finished polypeptide chain. Each of these steps requires the activity of a species set of protein factors in addition to the ribosome, tRNA and mRNA. Protein synthesis inhibitors are substances that disrupt the processes that lead directly to the generation of new proteins. They work at different stages of mRNA translation into proteins like initiation, elongation including amino acid tRNA entry, proofreading, peptidyl transfer, ribosomal translocation and termination. Thank you.